Welcome to week 7 of Bio 126. A few days ago, I was listening to a story on the radio about farmers in Guatemala crossing different varieties of corn to develop new plants that produced more corn and would be resistant to pests, drought, and they happen to be short in stature. You see, with the increase in storms and wind due to climate change, these farmers found that shorter plants didn't topple over as easily. These farmers were practicing a form of selective breeding. They were breeding plants with desirable characteristics such as drought resistance and short stature with other plants trying to retain those characteristics. There are some disadvantages to selective breeding, however. Number one, it can be really slow. New plant varieties can take 25 years or more of breeding to be developed. Number two, plants become less genetically diverse, making them susceptible to plant diseases. Remember the Cavendish bananas that we talked about back in chapter 1? Number three, selective breeding may result in the development of undesirable traits. But what if there was a way to be more intentional about developing new traits in plants? What if, during breeding experiments, there was a way to track the genes responsible for desired characteristics, so a farmer could predict which plants carried that trait before waiting until the plants matured? This could reduce the time needed to develop new breeds by 15 or maybe even 20 years. Better yet, what if there was a way to specifically insert the gene or genes responsible for a desired characteristic directly into a plant, so you could be sure that the plant would have that characteristic? That's what this week's work is all about. Plant biotechnology includes a number of techniques that help adapt plants to specific environments, needs, or uses. This week you'll learn about a couple of these techniques and investigate how they are used to address concerns such as drought and pest resistance, and how they can be used to help plants address human health concerns. The objectives for this week are to, number one, define the term plant transgenesis. Number two, discuss the impact of biotechnology on the agricultural crop production in the United States and worldwide. Number three, give examples of how plant biotechnology is used to improve plants and address issues such as human health, hunger, climate change, pests, environmental and economic concerns. Number four, you'll name limitations of conventional crossbreeding techniques as a means of developing new plant products. Number five, you'll describe at least two methods of modifying plants. Number six, you'll describe the use of TI from Agrobacterium as a gene vector. And number seven, you'll briefly describe the technology behind Arctic apples. For some people, this chapter is really interesting. After all, it describes the technology behind GMOs, and you'll briefly consider some of the controversies around and concerns around GMO crops. For other people, this chapter is sometimes not quite as interesting, but for those of you who find plant biotech not as interesting as animal biotech or forensics or some of the other topics that we talk about, I hope you'll find at least one thing that's interesting. If you have any questions about this week's materials, don't hesitate to contact me either using the Remind app or by email. I am here to answer your questions. Good luck!